Hello and welcome. In this tutorial, we will study the basic structure of the DNA and we'll study how DNA is coiled and supercoiled in the form of chromosomes. We all know that in non-dividing cells, the genetic material of DNA is in the form of thread-like structures inside the nucleus, and we called it a chromatin composed of the DNA and proteins. And DNA contain many genes, that are the basic unit of inheritance that are transported from parent to offspring and that control various activities. And how chromatins is condensed, and how DNA in it is organized in the form of chromosomes. Let's discuss. First of all, there are the five different there is the DNA, and there are the histone proteins. And DNA is wrapped around histone proteins, like this is our histone protein, and this is our DNA that is wrapped around the histone protein twice. And there are almost about 5 types of the histone proteins that help in wrapping of the DNA around them. And how it is helpful for DNA to be wrapped around a particular type of the histone protein is that histone has a negative has a positive charge, and DNA phosphate has a negative charge, so it is attracted to it and easily wound around it, and it is attached to it. And DNA, and which is wrapped around a stone in this manner, is called as nucleosomes. And there are 5 different type of histone proteins, and DNA is wrapped around the 4 histone protein, and fifth is linked between different types of such structures, and they are called linker histone proteins or linkers. Then there are super folding or higher level of foldings, that is further folding of the DNA and form supercoils. Like this is DNA, and it is coiled around histone protein in such a manner, and it is called nucleosomes. Then it is wrapped around itself and forming a supercoil. It is called solenoids. Then it is wrapped around and it is attached to the proteins called around the protein, that is called scaffold proteins, and it is forming a chromatin loop. Then it forms the rosets of the chromatins, that scaffold proteins. Various flower-like structures of the DNA are combined, and then they are supercoiled to form chromosomes. Then here is the highly condensed chromosomes. Then chromatins may be vary in different types, like there are the chromatin or the genes in the chromatins that are going to express readily, and they are called euchromatin, that is going to express. And there are other chromatin that are in condensed form, where the DNA is still in condensed, and they are called heterochromatins. And some of these genes may express later in life, and some may not be activated ever in one's life. Like if one has one has a color of eyes that is brown, then it will have definitely the genes for other colors, but they will also be remain dormant. And there are the many traits that are not going to appear, and genes are not going to express, and whole of our characteristics are merely because of 2% of the DNA that is in the form of non-condensed chromatins.